my friends. Welcome to the Fitomics podcast. I'm Dr. Cody Hahn. I'm the CEO here at Fitomics. And at Fitomics, we want to help you improve your health, transform your physique, and elevate your performance by providing science-based products and services. So we want to constantly integrate the latest scientific research in order to help improve how we do that. And with the podcast, we will be exploring the science of fitness. And today we're joined by Dr. Chris Vann, and we will be digging into omics. And I'm also joined by my esteemed colleague and dear friend, the one and only Dr. Daniel Lawson.
Thank you for the warm introduction.
Absolutely.
I am Dr. Daniel Lawson, and I am an exercise and sports scientist with Fitomics. It's a dream come true, truly.
Yeah.
And also a postdoctoral research fellow at the one and only Auburn University.
That's right, War Eagle.
War Eagle. And had a great talk with Chris on omics. Super excited about it. He's doing amazing work at Duke, and really hope you guys enjoy it.
All right, guys, with no further ado, here we go. Doctor, the one and only. I actually think there's another one because I Googled you and it came up with like some surgeon in like Florida. So I don't know if you have a brother.
I sure don't, but there are actually two other Dr. Chris Vanns.
Oh, wow. But you're the only real one in our hearts. Okay. And so Dr. Christopher Vann, esteemed postdoctoral scholar at Duke University in the, dual affiliations. So we have the Molecular Physiology Institute. Yes?
Yes.
And that's like your main activities happen in the Molecular Physiology Institute.
That is where I spend the bulk majority of my time.
Yeah. Yeah. It takes a while to do this sort of science, huh?
It does, yeah.
Yeah.
I have a much deeper appreciation for it.
Yeah, man. Well, I'm excited to dig in with you. And then we have Dr. Daniel Lawson with us here today. Truly the one and only. I do not know of another Lieutenant Dr. Daniel Lawson. Do you?
Not that I'm aware of.
One of a kind, man. Me, on the other hand, I'm just some schmuck. But hey, guys, excited to dig in here. We are going to get into some of the, Chris, it's like when we were at Auburn, we put that cardboard sign on that like closet that we would go into, and we would actually work in there. I think people thought that we may have been like making out or something. I don't know. It was probably sketchy. But we would actually work on our computers in this office, and we called it, remember, we wrote in like a Sharpie, it was like welcome to the rabbit hole.
Yes. It does not help that there was also a bed in there, but, we won't get into that.
Yeah. Well, and so we do want to get into the weeds on omics and some of the amazing research that you've done. Just a little background, right? We had the honor and privilege of working together at Auburn University under Dr. Michael Roberts, and in the Molecular and Applied Sciences lab. And man, I'm not sure I would have survived, buddy, without you. So thank you, first off, for being like a great friend and a great colleague. And some of the research that I was first author on, like you were right there with me, man. I really appreciate that, being in the trenches, and we did some really cool stuff. Learned a lot. And now we're here, buddy. We're here. It's special to be able to dive into the research that we've been able to do, going back shoot, almost 10 years, but
Yeah.
particularly right the last 8 years, 8 years, I would say we've been really focused on um, research and particularly in the areas of exercise physiology and then molecular biology. And you've really leaned in during that time period to molecular physiology, molecular biology. And yeah, dude, I'm excited for you to teach us today. Teach us. All right. So, first off, I really think it's good to give a little bit of attention here to omics, right? So, fit omics, we derive the term, fit is the root word of fitness. Pretty intuitive definition there, but we've defined that as like a set of attributes that contributes to the ability to perform physical activity that can be measured by specific tests. And then omics, there are different definitions, which I would really be curious to know if you had an even more concise definition. But it's really a sub-discipline of biology, right, that aims to
Right.
identify and quantify and describe the processes and you could say, depending on the form of omics, right, the proteins or RNA or metabolites or lipids that influence the form and the function of our cells and tissues. And so omics is kind of a broad field, if you will. And there are really sub disciplines of omics, the technique, and but there's only one fit omics. And so, to put those two words together, right, is it's a play on those words to where we want to use science, we want to use those techniques and a thorough understanding of someone's physiology to improve their fitness. And we believe that fitness is really worth pursuing, being fit. And that has, of course, health implications, but also performance and physique implications as well. I'm going to stop talking because we want to hear from you. But omics, what what is it? Uh, what do you think is a perhaps an even simpler way of explaining omics, and how does it differ from some of the more traditional methods that we have used to understand the molecular processes that influence, how we adapt to exercise?
So I think all great questions. I'm actually going to share some slides.
Oh yeah, buddy. Take us to the classroom, buddy. Take us. I'm awake. I just did like 20 intervals on the Peloton tread this morning listening to trap music, and there's quite a bit of lactate circulating in my system and my brain. According to George Brooks, Dr. George Brooks is being fueled right now by the lactate that I have generated.
Zoomed in a bit in present mode. Is that helping any?
No, Dan, do you see what I'm talking about? It's the
Yeah,
bottom.
it's in the bottom right.
Yeah,
It looks like a little dot that you can drag,
yeah,
plus and minus.
Yeah.
Oh, there we go.
yeah. Look at
All right.
us. Hey, we can do PowerPoints if we can do omics, guys. Everybody relax.
You know, interestingly enough, I feel like PowerPoints are the harder thing.
Right. It's just like I have a whole PhD just in navigating Bill Gates' brain. Um, so,
Yeah.
oh man, look at this. We've got it. It appears to be a muscle fiber. I'm seeing, it takes me back to Jurassic Park. We've got a lot of A's and T's and C's and G's here. We've got DNA, mRNA, protein. Help us understand this, doctor.
Right. So, before getting into omics, we kind of have to understand what we're talking about. And this is a super super broad overview of the central dogma of molecular biology. And to Dr. Cody Hahn's point, we do have a muscle fiber up top because that is my central focus is muscle. I think it's probably one of the more important organ systems in the body. And when you pair it with cardiac tissue, I would argue that striated muscle as a whole is probably the most important system in the body, followed very closely by the brain. But what we have is DNA, and going back to grade school, middle school, high school biology, DNA is a double-stranded in a helix type structure with 4 nucleotides, adenine, thymine, guanine, and cytosine, where A pairs with T, G pairs with C. And then as we move down to our most functional unit, which would be the protein, we have to have transcription, which would generate mRNA, which is a single strand, where our T gets replaced with U or uracil, but we still keep A, C, and G. And then from there, we undergo translation into a protein, and then those proteins are folded into functional units, which are really the basis of life. So this takes us through the entirety of the process. And it's worthwhile to mention the Nobel Prize in Chemistry this year was awarded to 2 AI platforms that predict protein folding. And that has been something that has provided molecular biologists with tremendous insight into primary, secondary, tertiary functions and foldings of proteins, and predictions of how they would look so we can better advance drug discovery.
Okay. So how about, this makes sense, DNA to mRNA to protein. If DNA wise and considering skeletal muscle, where is DNA stored, right? In the nucleus, and what kind of makes skeletal muscle unique compared to other cell types? Just kind of starting with that.
So one of the things that makes skeletal muscle unique is, one, it's post-mitotic. So it's not going to undergo mitosis and split. Although there is emerging evidence that fiber splitting does occur in humans, which is some really fascinating work that's happening at a few labs across the US and a few more in Europe. And that's something that actually Cody and I chased for a little bit while we were at Auburn. And it's really fascinating. The other thing that makes muscle really unique is it's one of 2 cell types that are multinucleated. The other cell type that is multinucleated are macrophages. So multinucleated just means that each muscle cell has multiple nuclei. So there's multiple little organelles known as the nucleus of the cell that house the DNA in a chromatin structure. And you have a euchromatin and a heterochromatin state, which is really whether or not they're kind of bound up and not able to be read for transcription, or they're more openly accessible so they can be read. And then the other fascinating thing that is worthwhile to note, the Wang lab at the University of Florida showed some really amazing data at a conference a couple years ago showing that when this transcriptional process happens, the T-tubule network, as well as microtubules within muscle, actually chaperone these mRNAs to various portions throughout the muscle, and they localize in certain regions, but not every transcript will localize to every region. They kind of have a signaling area where they're most needed and taken to.
So you're saying that there's now evidence, because you talked about the Nobel Prize this year in chemistry related to the prediction of how proteins are folded. And we could touch on why that is so important that they're not misfolded. But now you're saying that we've learned that the mRNA that are transcribed from DNA, not, I guess, to mention what you said about there's actual influence of the structure of the DNA within the nucleus, more epigenetic factors that can influence if the gene even gets transcribed. But you're also saying that post transcription, so we now have this mRNA, there seems to be a sort of directionality to where these mRNA are transported through,
Right.
did you say the T-tubule network?
The T-tubule and microtubule networks.
Okay. And so these are kind of like tunnels that these guys are just I don't know, walking. What are they doing? Just kind of like floating along. Have you ever been in there, Chris? Have you have you hopped on one and like taken? How do
You
we?
know, back in the third grade, I was in Miss Frizzle's class and jumped on the magic school bus, and
Shout
we
out to Miss Frizzle.
miniaturized
All right.
enough just to get into those networks.
That is where the freaking magic school bus was really going. All this freaking. I was wondering, man. Okay, so we're on the magic school bus. we are we're cruising through as an mRNA through the T-tubule network. And for our listeners, that's not don't cite us on that. We don't have direct experimental evidence that the magic school bus is actually the mechanism by which mRNAs are transported through the microtubule or the T-tubule network. But there is this transport process through this network that helps to get the mRNA into the intracellular space that it is meant to serve us.
Right.
Okay. And so, okay, we've got this mRNA. Let's like zoom in a little bit to the myofibril level. So we've got like our whole muscle fiber here, which is shown in the top image. And we go beneath the sarcolemma, the membrane. Now we're inside of the cell. And probably, right, Chris, according to our research, about 85% of that space is myofibrillar. And we've learned from some of Dr. Brian Glancy's work that it's more of a sort of lattice network, right? It's more of like a mesh, which is pretty mind-blowing at this point, right? We didn't know that before. So like, that's interesting. But there's, the myofibrils are the actual contractile apparatus. And within those myofibrils, we have the sarcomeres, the smallest, often used as the smallest functional unit of the skeletal muscle that has the myosin and the actin proteins, which is our final step here in the central dogma of molecular biology. So like we've got this mRNA that gets transported perhaps through the T-tubule or the microtubule network from DNA, right, into this space. And so now the mRNA is hanging out on the magic school bus, and we're going to, drop it off at its home. And what happens next? So I see translation here. Like, what does that mean?
So translation is the process of where that mRNA, you have what's called a ribosomal unit that sits down and reads the mRNA to create your functional protein. So at that point, you'll always the start codon, and a codon is just 3 nucleotides. Start codon is always A, U, and G. And I remember that because I was born in August. So
Nice.
Genesis.
Yeah, you got to. I have all kinds of weird mnemonics in my brain that some of them are not appropriate to share today, but we got to remember a lot of stuff for those dissertation, examinations, right?
Right.
So AUG is conceptually that's kind of like a signal, if you will. It's a code.
Yep.
It's logic. It's an algorithm almost, where it's just like, hey, this is the portion of the mRNA that the ribosomes, and you said there's a subunit structure to the ribosomes. So there's like a large
Right.
and a small. Is that right?
So there's a large and a small, but that can also be broken down further, which I don't think we need to get that far into the weeds.
Okay.
We can leave it as large and small.
All right.
Yeah, long story short, there's an 18S, 28S, and so on subunits that go along with this. But they will pull in, based on what they're reading at the codon level, they'll pull in the amino acids. So that AUG always codes for methionine. So methionine is the start of every protein.
Hmm.
And then from there, you'll pull in all of your other amino acids based on the triplicate, the codons there, the triplicate base pairs. And you'll form peptides and then proteins. And peptides are just smaller units of protein. And then from there, those will actually get folded into functional units through other organelles in the cell.
Like a myosin protein or an actin
Right.
protein or titin or desmin or nebulin and our
Right.
friends, vimentin. So, okay.
Or, energetic, phosphofructokinase, rate limiter, glycolysis.
So enzymes and proteins, which enzymes are proteins, right? One, generally
Yep.
speaking, right? Proteins are going to form the structure. But some of them are multi-purpose. Myosin technically is an ATPase. So it's a protein that gives structure, but it's also functional in the sense that it hydrolyzes ATP. But what we're really talking about here, right, is that eventually there's the translation of mRNA into protein.
That's right. Yep.
And so, okay, real helpful. The I think what would be good to touch on next is like how traditionally have we measured this, just like an overview, sort of each step, and then how is that different than omics?
Yeah. So you want traditional measures of protein or RNA, DNA, all of the above?
I guess, yeah, common methods, that we've used historically to assess DNA, RNA, and protein, and then how we've kind of built off of that to where we are now, and how, for example, like omics allows us to get more widespread information, higher throughput, more precision, that sort of stuff.
So the most common way that DNA and RNA are interrogated at a small scale are through PCR, polymerase chain reaction, which is a fancy way where enzymes are used to take an input of either RNA or DNA, and then you build libraries through amplification, which happens at various temperatures and through the enzymatic process that is most of the time when you're looking at PCR mixes, the mixes are proprietary. So we know we have a good idea of some of the things that are going into them, but not everything. So for instance, you might have an ELYSIS buffer when you're lysing a cell to even extract DNA. There's multiple different ways that you can do it. You can use an acid-based extraction, which is fairly quick. And one of those, for instance, is you can digest muscle in proteinase K, which will eat away everything. And then everything that's left in the liquid is just nucleic acid. That's all that'll be left. And then from there, you do an alcohol-based precipitation with ethanol and isopropanol to purify that DNA. And then you can extract it and you can measure the quantity, which would be in nanograms per a microliter. So we're talking very small volumes. Microliter, if you want to visualize that, is much smaller than a single drop of water. A single drop of water can be upwards of 1 milliliter. So we're talking 1,000 times smaller.
Mm.
And then within this, we have nanograms. So going back to chemistry, high school chemistry class, where you learned the different scaling of data, this is 10 to the negative 6.
Yeah,
And then, well, actually, nanogram is actually 10 to the negative 9. So, but micro is 10 to the negative 6.
right. Yeah, that helps put things into perspective on how small, we're talking here. You don't actually see the letters when you do and so I guess, we probably take for granted some of the steps here, but like we've taken a muscle biopsy. So we've actually taken tissue out of a muscle, and we have done sample preparation for the polymerase chain reaction. And you talked about, there's homogenization, you could say, of the
Right.
tissue sample. There's often centrifugation, right, to separate the sample into respective, components. And then we isolate, you say, the DNA, right?
Right.
And then you talked about purification. And so, one question that pops up for me, that I think would be relevant to get your thoughts on is like, how do we know that the DNA is stable? Like, have we damaged it when we do that process?
So, there's ways that you can measure the quality of the DNA, which will give you a proxy of if it's damaged or if it's not intact. And you also have to keep in mind you have double-stranded DNA, single-stranded DNA, you have double-stranded RNA. So when you're doing uh, the isolation and you actually look into the purity, there's multiple ways that you can do that. There's a wavelength base, so it's spectrophotometry. So what Cody and I did in the MASL, we used what's called the nanodrop, which emits light, and depending on the wavelength and how it's received back and the time that it takes, that gives you a proxy of quality on the 260 and 280 wavelengths. And that ratio, if it's 1.7 to 2, you have very good quality DNA. If you're below, you possibly have some contaminants. But there's also a 230 ratio that will give you a proxy of if you still have contamination from, say, guanidine salts, which are often used in isolations, or if you still have residual ethanol on there. Because your very last step is an isopropanol, a 100% isopropanol wash step that should remove all of the ethanol. But these numbers, and I know for a long time, we didn't pay attention to the 230 score. We only really paid attention to the 260, 280 ratio, but that 230 score is actually really important. But there's other ways that you can also measure, and they use similar technologies, but they give you an RNA or a DNA integrity number. And they're a little bit more of a specialized assay. So it's a little bit more work than just adding a drop onto a platform and running light through it. You actually have to resuspend these in a specialized buffer that allows, so what it does, the specialized buffer, is it will add various reporters that bind to the DNA, the RNA, and then it reads that. And when you're looking at REN or DEN scores, anything 7 or higher is outstanding.
Okay. That sounds fun. Made me want to go get some rubbing alcohol from my bathroom and see if I could do some DNA isolation, but I don't think that would work based on what you just described. Because I do not have a spectrophotometer on my desk yet. But there's this verification step that we've actually purified DNA, and then we would proceed with Dr. Carrie Mullis, I believe, is the who is credited with the PCR reaction. Apparently, this came to him from the ingestion of LSD-13 is the folklore. I don't know everyone, and we're not endorsing such behaviors, but there was a revelation on how that worked, where we could, you talked about amplification. So um, there's a step in PCR that allows, there's 40 cycles, right?
Depends on your primers, but yeah,
Okay.
rough estimate.
So we've got this polymerase chain reaction, polymerase being an enzyme, and so there's a reaction that allows us to amplify the DNA in the sample that we have prepped. And then you said that there are libraries of sequence information of genes that we can design primers for that would identify specific segments of DNA that would correspond to a given gene. And it's highly specific,
Right.
right? Because you said there's, U, A, C, and G in the mRNA. And so we could design primers that would be the other side, basically, of that strand, right?
Right. And it's important to note that depending on the application that you're using, so most people will do what's called RT-qPCR, which is a quantitative polymerase chain reaction. So generally speaking, when they're doing this, they're doing RNA extractions. They'll create an RNA library, and then before they thermocycle it, which is what Cody was talking about with 40 cycles is a common number to see. Some of the work that I do uses 38 cycles or 35 cycles. But you also put in the template, which is what your primers are, into that mix. And what you're really trying to amplify is that template. So when you actually analyze it, you're getting a proxy of how much of a specific transcript, your transcript of interest. So if we were wanting to look at myosin heavy chain, we would pick the common myosin heavy chain in muscle. And for slow twitch fibers, that's myosin heavy chain 7 or MYH7. So we would get a template for that. We would put that into the RNA. We would thermocycle it to amplify our signal. And then we would go downstream and do our actual PCR run, which would give us a quantification of how much myosin transcript we have in that sample.
Yeah, and so like each step of this PCR reaction, Chris, correct me if I'm wrong, but it doubles the material each cycle.
Yeah, roughly, yeah.
And so by running, 30 cycles, it really amplifies that signal so that we have all of these effectively like replications of that material so that we can be more confident about RNA abundance. Yeah.
Right.
And so, okay, we're still on one slide, everyone, and we're on step 2 of 3. And so, just to kind of zoom out for a second, we have this technique that we have used to amplify messenger RNA material in this sample, and that shows us how much of the gene is being expressed, and that's just one. That's in reference to one gene in this example, right? And then we would want to perhaps explore protein.
Right.
So,
And the most common way that this is explored is through a technique called Western blotting or immunoblotting, depending on where you are in the world and what you grew up calling it. But that is really, in a similar method, you do a digestion on the cells that you have, whether that's a whole muscle or if you're doing blood or serum plasma extraction or fat or whatever tissue you have. The most common way to digest protein is actually through a very widely used buffer called RIPA buffer. And with that, you would homogenize the tissue. Back in the day, Cody and I would sit there with these little micro pestles, and we would just sit there and grind away until it looked like there was nothing left in there. It was a slurry. It looked like a very opaque protein shake.
They're quite tasty. I actually sampled some of the samples, that we collected from our participants, and it has kind of like a salty, kind of umami flavor to it. So it was like a nice snack there in the Massel. So I'm kidding. Maybe I am. But okay, we've homogenized, right? We've broken apart
Right.
the tissue in this buffer, and then would we centrifuge it, right?
From there, we would centrifuge it. And what that's going to do is it's going to take your insoluble proteins and that's going to pellet those down at the bottom. And then all of your soluble proteins will be in what we call the supernatant, or the liquid that sits above the pellet. And we take that liquid off, and then we do some chemistry to it, some SDS page, which is a sodium disulfite. And then we color it blue with lamellin buffer, which we make our own in general. We got the recipe from the legendary Frank Booth, who,
Oh man.
worthwhile to mention, is the person who brought these techniques to exercise.
Yeah, right. Shout out to Dr. Frank Booth at Missouri, right? We are part of his lineage. We're very grateful for all of his wonderful contributions. And yeah, man, I guess, gosh, in the early 70s, Chris, right? He was publishing on, I believe AMPK was one of the early findings, right? AMP
So
kinase.
it was actually in the 60s because he did his first postdoc with NASA, and then did a second postdoc with the late great John Halsey,
Mm.
where he started to learn about these methods and started to bring them over to exercise.
Hmm. Yeah, and so I think Chris, right? He's probably the first, we could say, exercise physiologist, molecular exercise physiologist that really used Western blotting to study a pathway effectively. Right, some of that early work was on mitochondrial biogenesis, yes?
Right. Well, a lot of it was bioenergetics focused because that is what Halsey focused on quite a bit. But yeah, I think there were proxies for mitochondrial biogenesis. There were definitely proxies for the energy kinetics within muscle in general.
Yeah. Well, and so with protein immunoblotting, we've got this. I'll never forget Dr. Quindry, John Quindry, who's now at the University of Montana. Great guy, wonderful professor. He taught advanced exercise phys at Auburn, both one and two for me. Special place in my heart. But he explained Western blotting as it's kind of like we've got this protein, and imagine a forest of trees as opposed to a forest of whatever else a forest would be made out of. So we've got this, all these trees that start out further apart. And as you get deeper into the forest, the trees get closer together. And so as the protein travels through the forest, it, based on its size, it's going to get trapped in a certain portion of the trees in this case. And so, and what's pushing the protein in the case of Western blotting is an electrical charge. So we have this gel that has this grid, if you will, matrix. And perhaps we're in the matrix right now. Perhaps we are in a protein immunoblot experiment right now, moving through the gel matrix. And so we're moving through the matrix. You guys didn't think that was funny? Okay, cool. Um,
I think I'm
We
a giant protein moving through the matrix right now.
you are you're a large myosin,
Very large.
maybe even Titan, buddy.
You'd be at the 180 to 220 kilodalton range.
Right. And so we're pushing, and I think so right, Chris, based on the charge, we get this protein locked into this matrix. And then when the electrophoresis is over, we would take out this gel, right? And then what would we do? We would transfer it to something?
Yeah, I think it depends on the technique that you're using. So we've used multiple. Largely, if you're wanting to say, keeping with the myosin heavy chain um, analysis, what you would end up doing at that point is what's called a Kumasi blue stain.
Okay.
And then from there, you'll get a beautiful set of bands and lanes. And you can then quantify, so myosin's going to be sitting at the top of that, in that 180 to 220 range. Actin's going to be sitting more around the, if I recall right, the 70-ish kilodalton range. But you also have all these other bands that are in there. And included in that, you'll have troponin, tropomyosin. If you use a large enough gradient, you can get titin, you can get nebulin. But the gradient that Cody was mentioning, you can buy different gradients or different percentage gels based on your target of interest, which requires a little bit of background looking to find the specific molecular weight of that protein. So for instance, if you were only interested in myosin heavy chain, you could buy a standard percent gel of whatever would match that roughly 180 to 220 kilodalton range. And then you could do a very targeted analysis and only analyze that.
Mm-hmm.
But usually we came from a frugal lab where we had to stretch our money some.
Mm-hmm.
So we would often interrogate gels for multiple targets. So the gradient made way more sense for us to use. And we used what's referred to as a 4 to 16% gradient. So you'll be able to capture a lot of proteins in there. So that's one way to do it. You can also interrogate other things like phosphofructokinase or glyceraldehyde or GAPDH. Glyceraldehyde phosphate dehydrogenase, which is also involved in glycolysis. A lot of people also use that as a housekeeper, which not related to this. If you are finding yourself doing Western blots for some reason, it's worthwhile to note that the housekeeper that you use or the normalizer should be picked based on the tissue if you're going to do a gene-based or a protein-based normalization. Or you can do a Ponceau stain, which gives you a nice red stain that you can do whole protein loading normalization, which is actually required by most chemistry journals now. So just some food for thought.
Yeah.
There's students listening.
Chris, yeah, I think so. We're talking about Western blots right now, and before the first one we talked about was PCR. And just to kind of differentiate for the listeners, right, is PCR more looking at like the gene expression, and then Western blot is looking at the protein concentration. Is that
That's
correct?
right. Mm-hmm.
And then when we're looking at that protein content, and I have very little experience in a wet lab, but I do have some. My understanding when we're doing Western blots is we use a primary and a secondary antibody, correct, to identify that would target a specific protein of interest. And so that primary antibody binds to the protein of interest, and that secondary antibody is like a light, right? So when you're actually trying to look at it, it's kind of like a beacon of light that we can see. Is that correct?
That's right. Yep. And that's actually how it got the nickname is immunoblotting because that secondary antibody is generally usually an IgA, an IgG, or an IgM, which are immune antibodies.
Yeah, and that's with that, Chris, and we'd get those from um, a variety of species,
Right.
yeah, where the the organism is exposed to a given protein and then produces an antibody, quote, against that protein. Is that how you, and then we would actually isolate that antibody class from the organism and use that to identify proteins of interest in our own samples. Is that a fair overview?
Yeah, I think so. Yeah.
Okay. And so we've got the proteins trapped. We're looking we're out of the magic school bus, everyone. And so we are, we're going to transfer this to Chris, right? Is it PVDF? Yep.
Yes.
And so we've transferred it, and now the proteins are on this paper, effectively, a very special piece of material. And then we would incubate them, right, with antibodies, like Dan was saying. So we're going to put a solution in there, and we're going to rock it like a little baby, like a little baby protein. And we're just going to rock it overnight by hand. So we would actually sit in the lab with a lullaby and hand rock these PVDF membranes. Yes.
Yes. And for the record, they prefer Twinkle Twinkle Little Star.
As do I. And so um, we actually had a device, right, that we would put into a refrigerator, so it was a controlled temperature, and then the membranes would be um, moved carefully overnight so that the antibodies would bind to the actual proteins of interest, right? And
Right.
then we would take it and we would then use light exposure to capture photos of that emission of light. Yes. Uh,
Right.
and so based on that, we kind of get this image of density, really, of the protein in that specific region of the gel. And that would allow us to see how much of that protein was present in the actual sample.
Right.
Okay. So, and that's like an overview, right? But it's like that took all day. Actually, that took 2 days probably. And we're talking about, right, Chris, on one gel, we've got, I know there's variation in lanes or how many samples you can actually load onto the gel, but generally we used, I believe, 16 lanes. Yes.
So we started using I think they're 23 lane gels.
Okay.
But yeah, I've used as small as 8 lane gels.
Yeah. And so that's like if you had one subject, let's say, and we're doing a training study and we're looking at um, changes in myosin protein concentration in the muscle sample. And so we want to see normalized to the volume and the concentration of protein that we've actually put into the lane. So we're controlling for that. Um, we want to see if the concentration of myosin went up in the muscle cell over time. And so let's say we had 3 time points for one participant, and we're just running singles. If we wanted to do duplicate or triplicate for extra precision, like let's say we had two samples at pre, at mid, and at post for one subject, that's 6 lanes. And let's say we have 30 participants, you see, right? Like that might be as many as what, dude? 10 or more gels
Yep.
that we would use for one protein. I mean, you could get more out of it, carefully. You can, cut the membranes and make sure that you're looking for a specific, but it's a pretty laborious process. It's very delicate and you can make mistakes along the way. You have to be really careful.
Right. And the polyacrylamide is, it's pretty easy to tear. And
Yeah.
if you tear it, and if you have a minor tear, it's not a huge deal. If you have a tear that's really clean, you can piece that back together and still use it. But there are a lot of tears that happen that make that gel useless.
Right.
Then you have to run it again.
Yeah, and you're talking about like when we carefully transfer the gel onto the PVDF, like you can rip the gel and then
Hmm.
cry, um, and be depressed. So yeah, no, so
With
go
with
ahead.
this gel, um, it's like a net. Is that correct? When you're trying to filter the proteins down,
I think
or
you
am I
could
thinking of
probably
something
look at it that way.
where you have larger proteins and smaller proteins, right?
Yep,
And you're running
that's right.
electricity through it, right? Is that kind of a good way to conceptualize that? That's something that I remember being taught
It,
in the wet lab, or
it's
explained. That's how to.
Yeah, it's almost like a layer of nets that vary in size of the actual opening of the net. So you have like a wider
Yep.
net, and then the openings of the net get smaller. You can kind of imagine a bunch of nets being stacked on one another.
Right.
Yeah.
And that's when we're looking at the protein, right? And then you get your bands on this, I guess, sheet of paper, right? Is that's looking at the weight of the protein, right? So larger proteins get stuck at the top, and then the smaller proteins get stuck at the bottom. And then based on the where it lines up with uh, the kilodalton weight, right?
Yeah,
We identify that with a specific protein.
right. Yeah, I think
Okay.
that's spot on.
Well, we have clarified. Oh, I'm back. We have clarified the traditional methods, and that really is still a high level overview, right? And in future episodes, right, we will likely just really take apart um, each of those methods. Would be fun. But okay, PCR, Western blot, we get this protein. Cool. How does omics differ from this? Like, how would we actually, if I wanted to, because, like, wouldn't it be cool if I could take this muscle sample and with one method, with very high precision, um, could I just get all of the protein abundance instead of having to do all of the antibodies and the gels and ripping it and being on the magic school bus and crying? Is there a way that we could use omics perhaps to improve?
Yeah, so I think that gets us into what exactly is omics.
Yeah.
We
And
made it.
so, just a loose definition.
Hi, everyone. Just want to take a quick break from the podcast here if you're interested to have lab work done to let you know about the Research Labs Insider Service provided through Fitomics. Dr. Cody Hahn, could you talk more about that, please?
Absolutely, doctor. So, yeah, guys, it's a convenient way to order lab tests. And compared to other options, we often hear that folks save like 50%. The
Wow.
subscription covers draw fees and insider support. And so, yeah, it's a pretty easy way to get lab tests done.
Awesome. Massive savings on lab work. You can find out more at the Fitomics website. Back to the podcast.
Omics aims at the collective characterization and quantification of pools of biological molecules that translate into the structure, function, and dynamics of an organism or multiple organisms. And with that, there's multiple kinds of omics. There's genomics, which is done at the gene level or DNA level. There's epigenomics, which is where I have my primary focus in, which are things that modify gene expression profiles that are not attributable to the DNA sequence itself. Then there's transcriptomics, which is an interrogation of the RNA. So that's really getting into your gene expression. So these three play hand in hand. They're all nucleic acid based methods. Then we get into proteomics, and that's the translational portion of it. And you have multiple different flavors. It's like you're at Baskin Robbins and you're trying to figure out which of the 31 or 37 flavors to pick. You can do phosphoproteomics, you could do targeted proteomics, you could do peptidomics,
Oh,
all kinds of things. And then we
Trenbolomics.
can
Is that
Trenbolomics?
Trenbolomics?
That sounds great to me. Where can I find that at?
There we go. That's the soundbite right there.
Oh, God help us.
And then we can get even further down into metabolomics, which takes a look at the metabolites that are generated through all of these cellular processes. So you can get different acetyl-CoA fractions. You can get different acetylcarnitine fractions. So really getting into the minutia. And it's important to really think of, as we go around this wheel, we're going from larger. The only exception, so in size, would be genomics, transcriptomics, proteomics, epigenomics, and then metabolomics would be the smallest.
Okay.
And with that, we're interrogating large numbers of data. And just to kind of bring everything back in, again, the central dogma going from DNA, RNA to protein, with all of this together, we can look at genetic associations. We can look at how different polymorphisms, whether that's single nucleotide, multinucleotide polymorphisms, which is just a swapping out of a nucleotide or multiple nucleotides for a different one, how they can affect or influence what we're expressing. And then with the transcriptome, proteome, and metabolome work, we can really look at or begin to look at how all of this comes together to influence function. And really for our case here, are there different profiles of these ohms that are really signaling into a better powerlifting performance, a better marathon run? If you're doing an Ironman, is it influencing these different profiles, genetic, proteome, metabolome profiles? Are they influencing your performance in the Ironman? Is there a specific profile for all of these that Eliud Kipchoge has as he breaks the 2-hour marathon mark?
Wild.
What's the proteome look like when Ray Williams is taking 1100 to the house?
Right. Yeah.
Or Matty Rogers setting a world record in the clean and jerk or the snatch?
Uh-huh.
These are all questions those questions in particular are what really got me interested in this. But the health outcomes are equally, and for me, more so interesting. And how can we leverage this data to actually investigate that and how they relate to things?
So, yes, love it. So there's this interest around the ohm, right? Whether that's
Right.
the genome, the epigenome, the transcriptome, the proteome, the metabolome. And I like this definition of like omics is intended to create this collective characterization and quantification of pools of molecules that eventually will translate into the structure, function, and the overall orchestra, right, of physiology that happens that creates, movement and function and life, right? And so, okay, we've got these sub-omics. How would we, quote, do omics? So if I wanted to run an omics-based experiment and look at genetic associations, and one of the fascinating studies that you first authored, man, on like a genome-wide association study looking for unique genetic information that could help us understand why some people may grow more muscle in response to resistance training than others, right? You hear a lot, oh, it's genetics, right? Like this person has good genetics for X, right? They have good bodybuilding genetics, or they have good sprinting genetics, and it's like, well, what does that mean exactly? Like, is it the actual source code? Is it the sequence of the gene that they just possess? Shout out to mom and dad, right? Like they just have it, and then that dictates it. Or when we say, quote, genetics, is it actually epigenetic? Is there something unique about this person that would allow them to express genes more effectively, rather than like, do they not have perhaps a different code, a different genetic sequence? Maybe they're just better at accessing the gene and transcribing it into an RNA. And then it's like, well, maybe it's neither of those things. What if it's post-transcriptional? Is it related to how many ribosomes they have? And the function of those? Is it related to something else, kind of up the sequencing sort of process here? So if we just wanted to do like genomics, if we wanted to sequence all of the genes in a sample, maybe take us through that. How would how would we? Because it's very simple, right? Like it's, you just like take this stuff and like put it into like water, and then like drink it, chew it, spit it back out, put it in the thing you said, spectrophotometer. Like it's a rave. There's all kinds of different light, and then we get a graph.
Yes, and it's important that Kygo's music is playing during that rave.
Okay,
So,
yeah.
Yes.
Like,
and
yeah.
Kygo and Marshmallow.
Shout out to Kygo and Marshmallow.
Yeah, man. Yeah.
Yeah, before getting into that, I think it's important.
Hey there, listener. Taking a little break here to ask Dan about our online coaching here at Fidomics. Tell us a little about that, Dan.
Gladly. Well, so I've done the online coaching for the last 7 years, working with a wide range of individuals with different goals. And what makes Fidomics so unique in this space is we have a team of PhDs in exercise science and nutrition. And this team includes 2 registered dietitians. And so if you're struggling with weight loss or putting on a little bit of muscle, PRing on a lift, or maybe you got a race that you're training for, but you don't quite know what to do, we can help you. We all have experience personally training for those things, as well as helping people achieve those goals. And so we use advanced software tools to help with programming. And these are very user-friendly tools that you can access through your phone. This includes direct messaging to your coach, as well as calls. And so whatever it is that you're training for, we can help you. We got the tools and we got the expertise.
Right on. All right, my friends, back to the podcast.
Important that we look at the scale sizes of these data.
Sure.
So DNA is large. We have thousands of genes. When we get into the transcriptome, we have tens of thousands of genes. Generally, when you're doing a transcriptome-wide analysis, let's back up. When you're doing a genome analysis, so you do DNA sequencing, your data set can return upwards of 800,000 to 1 million genes. And you have to filter through that.
And
so that's quite a large process.
You do that manually, right? With a spreadsheet, you just sit there one by one and you inspect it yourself?
It depends on how quick you want to go gray.
Right.
And if we could probably see a little on the side, there's some gray going in.
Right.
No, so we code, well, I code a lot to do all of this. And there's various packages that are available to run in R, or Python, or Bash if you're doing it in the background of your computer. But really, this work hinges on the marrying of bioinformatics and biology. And without that, we would be sitting here for years upon years. Because if you sequence the human genome, I think Mike Roberts showed this in his genetics class that he taught at Auburn for a while. The very first human genome that was sequenced, they put it in binders.
Right.
And it took up bookshelves that encompassed an entire room,
Yeah.
with the first chromosome taking up an entire wall by itself. And keep in mind, we have 23 chromosomes.
Right.
And it wasn't until recently, so the genome sequencing project was actually just recently fully completed. This started a long time ago. Crick and Watson, who won the Nobel Prize for the structure of DNA, and they left out the great Rosalind Franklin, which interestingly enough, this is kind of a side note. So Crick and Watson got the Nobel for that, leaving out Franklin. The Nobel Prize for Medicine Physiology this year was given out for microRNA, which are near and dear to my heart.
Yeah.
They had a third party that was left out. Her name was also Rosalind.
Interesting.
And it was the spouse of one of the awardees.
Huh. What do we have against Rosalind, man? Rosalind's sounds like a cool name to me, much cooler than Cody. But, okay, so we've got this discovery of the structure of DNA. You talked about, was it Francis Crick and James Watson? Yes, just like the helical structure. And now we are looking at and then I think, Chris, like you said, I think it's been at least 25 years, right, since we started the Human Genome Project.
Right. And we had 80% done in the early 2000s, but it took us almost 20 years to get that last roughly 20%. And so the most recent update to the human genome was just pushed out, and that includes the sequencing of the X and Y chromosomes. And it's also fairly important to keep in mind the Human Genome Project was done on an N of 4.
Yeah.
So 4 participants had whole genome sequencing done.
But if you read the internets, the interwebs, that's a not enough sample, buddy. Nope, not going to cut it. We need at least 4 million for it to be a high quality study. So yeah, we can.
Where do we sign up to do this?
Yeah.
Great.
Where can our listeners go?
Yeah.
Drop a link.
Yeah. So Chris, we wouldn't use, well, tell us, like, it wouldn't be PCR, right? We would use nowadays to sequence the entire genome. How would we go about that? And perhaps this could bridge to the genome-wide association study that you did, looking into, you want to take us through that?
Yeah, so data scale wise, we have hundreds of thousands of potential genes. When we go through translation, we can narrow that down to functionally 60 to 100,000 genes if we're collapsing all variants into one. And then at the protein level, we have tens of thousands to hundreds of thousands of functional proteins when we look at just standard and modified proteins. So with the DNA sequencing, you would do similar methods to get your DNA sample, but then there's a couple of different avenues that you can take. You can do a SNP array, which arrays are these little chips that you would put your sample on after you hybridize that with chemistry, oligo-based chemistry. And then they're read, and what you have in your sample is matched up to what's in the gene array. When you do sequencing, though, what you have is what's called a flow cell. And in this, you have all of these different lanes and all of these different sequences within the lane. And you can think of them as just floating there. And it could be like an A, C, T, G. And then with the sequencing, when you apply your sample to it, that binds based on complementarity to what is in there. And then that can be quantified through light spectroscopy. Usually most often using red and green signal. And that's how we get our genomic, and really that's also similar to how we get our transcriptomic data. And it might be worthwhile to ask yourself, well, how does that relate to exercise? So a lot of times when we're looking at exercise, our questions of interest, and we had just mentioned a few. What's making Ray Williams squat 1,100 pounds, or making Kipchoge run 2 hours for 26.2 miles? Which, a sidebar, I don't know if any of you guys have ever tried to run his pace for even a little bit, but your man made it about 45 seconds at his pace. It is fast.
Yeah, what's the mile pace on that, man? I could do some mental math here, but that's booking it for 26.2 miles in under 2 hours.
Yeah, so even if we do just simple math, I'll break out the old iPhone calculator here. Shout out Apple. Love them.
So it would have to be, 13 miles an hour, right?
Yeah.
So
So
Yeah.
you're booking.
yeah, that's wild.
Yeah, it's like a sub 5 minute pace, right?
Oh yeah.
That's
Yeah.
yeah,
I think it's pretty dang close to, it's just over a 4-minute pace.
yeah, man. Wow.
Oh, that low. Yeah.
Yeah.
But we can take those traits and, we can take it more clinical variables, VO2, insulin, anything like that. And if we measure a large sample of people, we will almost always get a plot that looks like this. This is just a waterfall plot. So you'll have some people who decrease insulin sensitivity. In this case, some people that increase insulin sensitivity. So there's just wide variability. And this is what we've really started to come into in the exercise literature terminology, calling them responders or non-responders. And I think it's pretty important to clarify, in a given study, we're testing a certain protocol. So for resistance training, and the study that always immediately comes to mind is Dr. Hahn's dissertation, where people got absolutely destroyed, starting with 10 sets of 10 on the squat, the stiff leg deadlift, a pressing variant, whether that was bench press or overhead press, and lat pull downs. And then over a period of 6 weeks, we progressed them up to doing 32 sets of 10. And keep in mind, that's for each exercise at the end of it. And we even saw something similar to this, where some people got smaller and some people got very large. Even with my dissertation, where we did unilateral leg training with high load on one leg and high volume on another, we had one participant whose high volume leg grew 30% in 6 weeks. It was one of the most interesting sights I've ever seen in my life, where he came in very balanced, and at the end of training, it was visible that one leg was way bigger than the other.
Yeah. Yeah. Just the eye test on that one was wild. Yeah.
So what we would generally do after we identify something where we have a heterogeneity of response, which is what this really depicts, is there's not everyone responds equally, really is what that's saying. We can then look at molecular mechanisms. So the example given here is total fat oxidation, which is associated with insulin sensitivity. And we can see that depending on exercise intensity, you might have different responses, depending on group assignment, or this could be sex, or it could be age, or, any number of variables. And then from there, where the integrative omics or multi-omics comes in is we would actually start to look at the genetics or the transcriptomics or the proteomics, the metabolomics, et cetera. And maybe we don't just want to do that in muscle. Maybe we want to do it in circulating plasma, adipocytes, or fat cells for lay terms. Maybe we want to look at the brain. I'm not suggesting we do a craniotomy and just take a plug of your brain, but maybe we're doing it in mice and we start to segment out the brain to see what's happening there, or maybe the liver. And then from there, with all of this data, what we can do is we can start to look at what does the endotype result in? And we can marry together the metabolome, the proteome, the microbiome, which is one that we haven't talked about. And full disclosure, I'm not adequately equipped to talk about the microbiome, but the epigenome. And we use the bioinformatics and the machine learning to get at least classifications of people. And that's how, using this type of data, we can really start to dig into, provide very precise recommendations for different subpopulations of the global population, really.
So, okay, this is a nice visualization here. We've got some variable of interest. In this case, you've mentioned someone's VO2 max and perhaps relating that to their insulin sensitivity. And so we do an exercise intervention, let's say, and we look at how much someone's VO2 max changed and their sensitivity to insulin, which we could use like a HOMA-IR score, for example, and look at insulin and just glucose and calculate that. But we've got a measurement of, and you could, of course, do like an oral glucose tolerance test and more or less sophisticated methods, but we've got insulin sensitivity, some kind of change in that. And ideally, for metabolic health, right, we want that to improve in this example, but we see that there's variability in how people respond to a given dose, a given intervention of exercise. And omics could help us to understand the reasons, the underlying molecular happenings that may or may not have influenced their response. Right?
Right.
And one of the more intriguing things about omics, just to kind of circle back to some of the previous discussion, is that we can get a lot more information in relatively shorter time with higher precision using sequencing methods, right? Using omics to be able to learn more expeditiously, you could say. It really can help to knock down, I mean, in general, right, if we can improve throughput and precision, that's a great option.
Right.
Right.
But it's also important to take note of when you're doing an omics approach, whether it's single ohm or marrying multiple ohms together, these are largely relativistic measures. They're not absolute. So a lot of people will try to compare PCR or Western blots with proteomics or transcriptomics, and they'll see, maybe on PCR you have a gene that's going up, being upregulated. But on the transcriptome data, when you take into account everything that goes into the processing, you might see no change, not as high of a change, or sometimes maybe even a downregulation or a decrease in the expression of that gene. So the interpretation of the data doesn't always match what the single single unit interrogation is showing us.
Okay. Well, and I think it would be helpful to spend some time here just clarifying. Let's just stick with muscle. We spent a good amount of time looking at traditional methods, PCR, Western blot. You explained flow cell, and you talked about kind of this mysterious floating process. If we have muscle, and instead of using Western blot, I wanted to look at all of the myosin proteins. So let's just say 1, 2A, and 2X, and add in some other proteins of interest. Why does omics, why is it more appealing than Western blotting?
So with Western blotting, you get information on a single target of interest, or at best, maybe you can probe 2 or 3 targets if you run the right gradient gel. If your interest is in contractile proteins, there are several myosin heavy chains within muscle. There are several actins within muscle. And then we also have your titans, your nebulins, your desmins. If we do proteomics, now we can get a relative measure of all of those in one shot.
Yeah.
So sometimes if your question of interest research wise is on a single gene or maybe 3 or 4 genes and 3 or 4 proteins, doing an omics approach is probably overkill. And you're going to spend a lot more money to do that. But if your target of interest is several pathways, and staying with the muscle theme, maybe we are really interested in muscle growth pathways, with the most classical being the PI3K, AKT, mTOR signaling axis. Within that, the broad overview is, from that point, you would want to interrogate your IGF receptors, which sit on the cell surface. You would want to interrogate PTEN. You would want to interrogate your IRS proteins, which are insulin receptor substrates. And insulin, IGF-1 is sometimes used interchangeably with mechanogrowth factor. But then from there you have PI3K, you have AKT, specifically in muscle, I believe it's AKT3. But don't quote me on that. It's been a while since I've looked at that literature.
I think you're correct.
And then you have mTOR, but mTOR doesn't act alone. mTOR forms a signaling complex with Raptor and MLST7 to form the mTORC1 complex. But there's also an mTORC2 complex. Maybe you're interested in that. And then from there, you have downstream signaling of P70S6 kinase, 4EBP1, not necessarily in that order. But then ultimately your outcome is muscle protein synthesis. That's a lot of proteins, and that's just the broad overview. There's several more that are involved in that whole signaling cascade. So proteomics gives us a better shot at capturing what's happening there. And generally with that, you have a couple of different ways you can go to normalize data. You can, again, use a housekeeping protein as a normalizer, something that in muscle absolutely doesn't change no matter what you do with it. You can pump it full of, let's say maybe you put 500 mgs of test in there, but that protein doesn't change and all the other proteins do. Or you put, I don't know, 500 mgs of estrogen in there and you want to see what happens. You find a protein that doesn't change and you can use that as a normalizer. Or you can use other normalization methods for that, which would normalize to a whole protein library or something like that. And then it gives you a relative change with your intervention. So if we stay with testosterone pre versus post testosterone administration, you would get a proxy of, well, myosin heavy chain 1 was, 7, just pulling an arbitrary number out. But after 6 weeks of testosterone administration at 500 mgs, now you have 14. You've increased your myosin heavy chain 1 twofold.
Mm-hmm. Well, and as far as the method to generate that data, the sample prep you described is similar to how we would prepare the sample for Western blotting or protein immunoblotting. I know that there's a little bit of variation there, right? But
Right.
there's
There's multiple different ways. You can use a urea-based protein extraction, which is what a lot of proteomics cores used. You could use a hydrochloric acid-based extraction. Something that we generated in the Massel, the Roberts lab, we generated a contractile and non-contractile protein isolation method that uses a substrate called spermidine to actually solubilize the contractile proteins, and then we fractionate those out. So we would do a similar method to normal protein extraction, and we'll pull off the soluble proteins, and then we resuspend the non-soluble proteins, which is our contractile units, and then we would homogenize that again, and the spermidine allows the dissolving of those, and then those can be done, be submitted for proteomics, which we've actually done multiple times. And with high volume training, for instance, what we found in Cody's project is an increase in enzymes, proteins related to glycolysis, which was a fascinating, very unique finding. And then, a year later, we published a paper with high load training where we did proteomics. And we saw a completely different signature where we actually saw an increase in certain contractile proteins, the troponin isoforms. I believe it was troponin M and troponin T that increased. But we also saw some mitochondrial related proteins that increased. So it gives us the ability to look at a large variety of targets with that. And proteomics in general is done on a mass spectrometer, usually either a liquid chromatography or a gas chromatography mass spectrometer. And it's usually a tandem. So it's you'll see it annotated as LCMS-MS. So there's actually 2, which would signify that it's a quadrupole usually. And then those use, similar to what Dan was mentioning earlier, with a net, if you will. It's like multiple nets where your proteins have a charge and they're being either pulled up to one net, pushed down to another net. If they don't match that, they get pushed to the next. And it goes like that. And that's how they're captured and quantified. And you're looking at the very specific peaks of those proteins.
Yeah, based on their mass, right?
Right.
Yeah. And so, okay, and we have this visual representation that we can quantify how much of that protein is present in that sample, similar to how we would use protein immunoblotting, but now we've captured a lot more protein information, right? And so that is particularly intriguing for exploration around what we're talking about here with heterogeneity and response, right? Like us three, we do Operation Tactically Thick with Dan on the app. Little plug there for the Phenomics training app, right? We run this program before and let's say 12 weeks after, we do a body comp test and perhaps like a VO2 max test, and we all see a different response, right? And so, okay, why? And there are certainly things like sleep and nutrition and lifestyle, stress, those sorts of things for sure that influence the response. But is it actually physiological, right? Is there something going on at the protein or gene level or translation level that influenced our responsiveness. And omics can help us to facilitate a sort of deeper understanding, really, of this molecular milieu that could help to understand if that was a better explainer of the response than other variables of interest. So if we controlled for diet, let's say we control for sleep, and we're never stressed, right? So like we're all chill, and we're controlling for all of that. And we were just really interested in what physiological mechanisms were at play here. Is there good evidence for genetics in using genomics or like a genome-wide association study? Do we have any insight from your research on a gene or multiple genes that are different between individuals that respond really favorably, that grow more muscle from resistance training versus individuals who do not?
We do actually. So jumping into that a little bit, we did a genome-wide association project, and I actually call this a baby GWAS because it is by far underpowered. In collaboration with Stu Phillips's group at McMaster, who's a great mentor and colleague and friend.
Yeah, shout out to Stu.
His former PhD student, Rob Morton, who a lot of your audience may or may not have read certain papers, meta reviews from him on the optimal dose of protein, for instance. But they did a training study that was similar to our good friend Brooks Mobley's dissertation in untrained individuals. So Stu sent us quite a few muscle samples, and we had quite a few that we were able to rescue, if you will, out of OCT, which is a compound used for doing microscope work, histology. And I did a DNA isolation on them. Initially, we had in total 120 participants with available tissue. And keep in mind, a typical genome-wide is considered low-powered if you have 1,000 participants. Ideally, you'll have 10,000, 30,000, upwards of 100,000 participants. Because when you're doing a genome-wide association study, what you're really trying to do is you're trying to extrapolate the contribution of a certain genetic variant or a certain gene to the entirety of the population. And if we think about the global population, we're now over 8 billion people. So you would also, in a perfect world, ideally have that sample represent the rough estimate of what is the Asian population, the Indian, African, Middle Eastern, European, Russian, all the way to South America, North America, and have your population be roughly an equal representation of all of that, which is hard to do in a lot of cases. So what we ended up with was 120 participants from Canada and Alabama, which ended up being predominantly white males. They were all males specifically, so I'm sorry, ladies, we left you out. Not intentionally, though.
Right.
And then I did all of these DNA isolations using an acid-based extraction with proteinase K. And then we submitted these to a company in Atlanta, Georgia, where they did a SNP array, the Axiom, the Axiom whole genome array, which looks at single nucleotide polymorphisms, or where you have a single nucleotide that's switched out for another one. And we found some really interesting stuff initially. But as we cleaned the data and we started going through everything, what ended up coming out.
Hey there, listener. Thanks for making it this far in the podcast. We told you a little earlier about our Research Labs Insider Service and about our online coaching. Dr. Dan, tell us about the Fitomics training app.
Absolutely. Well, one of the things that I get asked a lot is, Dr. Dan, what should I do to improve my body comp or build muscle? And that's what's great about the Fitomics training app is it's a very valuable resource that contains a variety of different training plans built by us through years of experience that are readily available and a very user-friendly experience within this app. And you pick a plan and you follow it, you log your sets, reps, exercises, and you track your progress over time. And it also functions as a great workout log to just, if you're struggling keeping track of what you've done previously and maybe you know what to do, but you just want to log your weights and easily access that, it's a great tool for that. And there's other resources that are available in the app, and we're continuously adding new training plans that are coming out, and it's a great resource. And for less than $1 a day, you can access all of that, and you can also try it out for a week free.
Nice. All right, back to the podcast, my friends.
And this is called the Manhattan plot, where it takes every gene that is on the array by chromosome. And what you can see is the spread of the data here. And this line that's at 5, so we take a p-value, and then we log 10 transform it. So this p-value is actually very, very small. It's 10 times, or 1.0 times 10 to the negative 5 overall. And we identified 2 single nucleotide polymorphisms, RS4675569, which is an undocumented gene. So that's also something to keep in mind looking at genomic work, is there's still undocumented genes. And then we found GLE3, which in the genome is marked as RS10263647. And it's important to note that this is an intron variant of GLE3. So presumably it actually doesn't code for a protein. Because when you go undergo transcription, introns are spliced out of the genetic code, and exons are what are either pulled through or recombinated into different ways. And then that's what actually undergoes the transcriptional process. So with GLE3, we found a pretty strong association with muscle fiber cross-sectional area, mean muscle fiber cross-sectional area specifically. Whereas if you have the T to T genotype, which was only 15 of our participants, functionally you really had no change in muscle fiber cross-sectional area. So that's homozygous negative, if you will. If you were heterozygous, meaning you have a copy of each, so you have a T and a C, we saw that after 12 weeks of resistance exercise, you had a pretty significant growth. But if you were C to C, homozygous positive, you had more growth overall, which the median is a little bit lower, but the overall spread of growth responses was higher. And then what I'm not showing here is further extrapolating that out. We also saw in a very similar pattern, satellite cells increased in a stepwise manner based on your genotype. And satellite cells are muscle-specific stem cells that are very, very important in muscle growth responses as well as muscle regeneration responses. So if we keep in mind every time that we exercise, whether it's running or jump roping or resistance training, riding bike, we do micro damage to our muscle, and we have to repair in order to at least return to baseline, but ideally improve past baseline so we can continue to have that type of improvement. This gene seems to be playing a role in that. But keep in mind, we had 120 participants at start, but what ended up being included was only 107. So very small sample size. So this is work. We ended up publishing this in FASEB, which arguably is probably the best molecular biology journal. It's certainly one of the best. And it was kind of a career milestone to publish a paper there, especially as a first author.
Mm-hmm.
But this is a small project that will be followed up on in the future in larger sample sizes. One thing that's of benefit is there are growing sets of biorepositories. So the NIH, when you do work with them, they have a system called GEO, which is the Gene Expression Omnibus, that you have to upload all of your raw data to. It's de-identified, so no one knows who's in each study, but it's essentially a repository of genomes and transcriptomes, proteomes, etc. Then you also have other samples like the UK Biobank that have similar things. So we can expand this tremendously and subset out for studies that looked at muscle growth in this case, if we want to validate, which is something that we're planning on doing. We can also start to do some gain of function research where we start to manipulate this GLE3 gene. But what's important about GLE3 is it's in the hedgehog signaling pathway. Hedgehog signaling pathway was named after Sonic the Hedgehog, actually.
Right on.
Because if you go into the modeling of the folded protein structure, going back to the Nobel Prize this year, the folded hedgehog proteins look like little hedgehogs, and that's how they got named. Just a little anecdote there. But
it actually is Sonic the Hedgehog, and he is actually on and off the Magic School Bus in the T-tubule network. That's what's actually happening, folks.
It's the crossover everybody wanted.
oh, yeah.
So, okay, and when we see RS and this ID next to it, and you've got the GL, you could call it the GLE3, GLI3, that RS ID there, that is a single nucleotide that is different in how many people here. Like I understand we've got all of our chromosomes on the x-axis, we've got effectively probability on the y. Probability of what exactly? Because I see these two dots are over 5. It looks like that's a threshold. One is an unknown SNP. But what are we saying there with that, the GLE3 gene? It's improbable that it would be so highly expressed by chance as far as its relationship with muscle growth, or kind of unpack what that really means, why it's so much higher.
So what we're really identifying here is the specific genotype by outcome. So I wouldn't look at this as gene expression, but more or less in a specific set of genetic code, which in humans, we're looking at I think it's 6 billion bases in DNA.
With a B. Yeah, billion.
Yes, absolutely. Just a fraction of Elon Musk's net worth.
Yeah.
But,
Right. So we've got all these bases, the nucleotides, and there's a pair, there's a sequence, and we're saying that a SNP, a single nucleotide polymorphism, is a one letter or nucleotide difference in a specific gene, right?
Right.
And
So
yeah, take us through.
Coding wise, the way that this is all done is you have an outcome of interest. In our case here, it was fiber cross-sectional area. and satellite cells, which I'm not showing. But you have your outcome of interest, and that's a continuous variable. And then you have all of these inputs, all of the SNPs, and it's a quasi-regression style analysis that goes through all of that data, corrects for multiple testing hypotheses, and then only these two met that threshold. And this is a lower threshold than most. In fact, generally what you would see on this log 10, negative log 10p is 0 to 10, with 9 being that cutoff. So we loosened our criteria for significance intentionally because our sample size was so small. But if we had, 10,000 people in here, we would have kept the 9. So it'd be 1 times 10 to the negative 9 instead of 1 times 10 to the negative 5.
Okay. And so we're basically saying here that it's improbable that we would see a relationship between the presence of this single nucleotide polymorphism and change in fiber cross-sectional area.
Right. Mm-hmm.
So it's unlikely that by chance alone we would be observing this SNP in these participants, the relationship of this SNP with muscle growth effectively from resistance training. And then you said that this gene seems to, and folks, right, like this is one SNP. There was nothing else other than this unknown gene, this unknown SNP. There was nothing else that appeared to give us confidence of such a relationship, right? Of all of those other potential SNPs like ACTN3 and ACE and myostatin are some that come to mind that others have presented maybe would be different. We might see some SNPs in those genes for muscle growth.
Yeah. I have another slide that we got into that too. I'll have to switch the slideshow though.
Okay. So, well, before we switch it, you think that why does this make sense physiologically? Like, what is GLE3? You said it's not protein encoding. And in fact, shout out to our friend, Dr. Matt Romero. I recall from our glory days at Auburn, got really into, and our friend, Dr. Shelby Osborne, were interested in, and even Dr. P.D. Mumford, right? It became a real interest in, quote, junk DNA, which we've moved away from that expression, I think, at this point. It's DNA that does not encode for protein. And in fact, Chris, right, that's most of our DNA. We aren't sure that it actually does encode for protein.
Right. And that's why on that very first slide, when we were looking at the scales of data, the DNA is 10 to the 4 or 5 instead of 10 to the 7 or 8, because most of the DNA ends up falling into these intronic regions, and it's only the exons that are undergoing the transcriptional process.
And in this case, without getting into like epigenetics here, in this case, GLI3 or GLE3, why does this make sense physiologically?
So GLE3, like I had said, it's part of the hedgehog signaling family, which is part of the it's part of the developmental and regenerative pathways. And that's not just in muscle, that's in a lot of tissues. So it could very well be, and I'm spitballing here. So there's no evidence to back this up. This is part of some of the future work that I'll be doing, but to follow up on this. But it's possible that this intron variant, keep in mind, introns aren't going to be transcribed into an mRNA and ultimately a functional protein, but perhaps this intron variant, and we don't know where it lies on the gene, right? So it could be in a promoter region. that maybe in a promoter region, we'll have to get into a little bit of epigenetics with that, but a promoter region is upstream of the actual gene, and that allows the signaling for RNA polymerase to actually start functionally working for transcription. So it's possible that maybe this TC variant, where TT results in lower fiber cross-sectional area changes, maybe it doesn't allow for, or doesn't allow as rapidly even for GLE3 to be read.
Hmm.
Or maybe if you have that C, that cytosine come in, maybe now you're having just a little bit more accessibility to the actual gene itself. And if you have the C to C, you're having a lot more accessibility to the gene itself.
Hmm.
That's just one possible hypothesis. And it also could be, so that would get at chromatin changes, going back to the DNA being in the form of chromatin in the nucleus. But you could also have the potential for maybe if you have the CC, now you have in the intron area, a CPG island, which gets into DNA methylation. And we don't know the answer to this quite yet again, but maybe now you have the ability to have hyper or hypomethylation, which would again affect how the gene is being read, whether it's being turned off or turned on.
Hmm. Okay. Well, and you mentioned like a promoter region and RNA polymerase. So maybe an analogy would be helpful of like it's kind of like a runway almost for this molecular structure that eventually results in transcription into mRNA or into, you mentioned before, we started recording the 12 different kinds of RNA, but for simplification, messenger RNA, let's say. So the promoter region is like a runway that can affect, and the speed at which that structure perhaps moves on the runway could be influenced by the base, the sequence of bases, almost like a speed bump in the runway, or not, like a nice smooth runway. So perhaps that could affect the rate of translation, and eventually that could manifest as a difference in the rate of, transcription, and that eventually, if there's more RNA, mRNA present, then across a certain amount of time, well, then it would be more likely that those mRNA would be translated into proteins.
Yep.
And so that's kind of one hypothesis here is this SNP could be affecting the eventual expression of relevant genes that would affect muscle growth in response to resistance training, but that we don't have data. That is a guess, right, at best. So you're going to be
Right.
looking more into this. I'm really struck by the lack of findings. I remember, when you started this experimentation, just with all the reasonable contention around like, well, it's genetics, right? Like we've all had that friend that it seems like they can just look at a dumbbell and they get like an 8-pack, you know, and it's like cool. They kick the wall 4 times and they have Tom Platt's quads, but it's like okay, there's variation in responsiveness. Um, the that seems to be genetic is what most folks would kind of point to that differentiates responsiveness. But in this study, looking at 107 was the final sample size.
Right.
We really saw one SNP of one gene that we would be confident, with limitations of the study in mind, saying like perhaps something's going on here.
Right.
And my hypothesis was that we would see more, we would see more interesting findings. What do you think about that?
So I think like a lot of things, if you take every gene that's ever been identified that goes along with a certain phenotypic trait, it almost always comes down to if you do the averages, right? We all regress to the mean. Any given phenotype has roughly a 50% genetic component and a 50% environmental component. And there's quite a spread there, right? So if you look at some of the developmental diseases, those are going to have a lot higher of a genetic component. But if you look at some of the other things, maybe they have a lot more environmental component. And that's where I think epigenetics really comes in. And epigenetics in general, it's actually an incredible question. I think it's very important, particularly as we try to move to more of a precision framework for exercise recommendations, nutrition recommendations. When we get this data, if we build these repositories, and keep in mind, and Cody had just mentioned MotorPack, which when it's finished will be the largest exercise trial that's ever been done. And it has a plethora of information, genomic, transcriptomic, proteomic, metabolomic, all of these different measures across several thousand participants, both male and female, which was really important. And then we also have untrained as well as trained. And then that's further classified down into previously resistance trained, previously aerobic trained. And you have different arms of exercise. So what's happening at the molecular level with resistance exercise, what's happening at the molecular level with aerobic exercise. It's going to give us a ton of information that we don't have right now. But there's also, with every study, there's limitations, of course. And one of those for MotorPack is there's no dose response relationship. It's a single type of aerobic training. It's a single type of resistance training. So some of the other work that's been done on this, the STRIDE studies, which right now are the largest completed exercise trial. And there's 3 STRIDE cohorts, largely interrogating aerobic exercise of various amounts and intensities in men and women across a wide age range.
And Chris, shout out to Dr. Krauss, right? That is
Yes.
really at the Molecular Physiology Institute, your mentor. That was
Right.
pivotal in the STRIDE studies. Yeah.
Yes. So Bill Krauss, who is here at Duke, he is the PI, the main PI of the STRIDE studies, and there's a whole slew of people like Chris Slintz and Joe Humard who have been integral parts to this study. And these studies have been ongoing literally for the last 20 plus years. So there were 3 cohorts of participants. We have in total just under 1,000 participants. We've done genotyping on everyone. We're working on transcriptome, small RNAome, DNA methylation for every participant. And that's going to give us a picture of what's the dose response look like? And how can we further stratify this down with the dose response? And then we can also leverage other studies like the famous study, which Priscilla Clarkson did. Monica Hubel helped out with that. And Monica's another great friend and colleague. Where they identified certain genes, some of them Cody had mentioned, ACE, actinin 3, as really important for muscle in general. And then you also have the heritage family studies, which were probably the initial classical studies that Claude Bouchard was the PI of, and Bill Krauss was involved in that. But you also have people like Mark Sarzinski at South Carolina who've been involved. Where they identified several genes that at least explain part of the variability in response for VO2 or aerobic fitness. So it's part of it is a time game and getting all of this data together and getting everything analyzed in a proper way so we can get this information. So then we can start to disseminate it in an appropriate way. And you got to figure it wasn't until late last year that the American Heart Association endorsed resistance training as a meaningful exercise modality for cardiorespiratory fitness.
Right.
But Mike Stone showed that data over a decade ago with a 10% increase in cardiorespiratory fitness with 10 weeks of resistance training.
Mm-hmm. Yeah. Well, and so there are a lot of great scientists working in this area, and with the rate of innovation at this point, we're in a really special time to gain insight, to help people, right? Is kind of how I look at it, as like, this is not, an effort to collect everyone's genetic information to develop clones and, create advanced weaponry to harm people, which is, I think when people think genetics, it's like, it's concerning like that, but the intention of this work is to get information so that eventually we can affect practice, right? We get, as far as I can tell, one trip on planet Earth, and would like to make the most of our time, right, in this experience. And so we want, people's time is precious, and effort and resources. And so it's like, if you're going to invest time and effort and energy and money into exercise and nutrition strategies, then, man, ideally we're not wasting any time or any of those resources. And that's kind of how I see this sort of research helping, to address that. Would you agree with that?
I do, yeah. And there's, doing genetics research, it's not just limited to exercise responses. We are now at a point to where we have genetic-based treatments for sickle cell.
Yeah.
We have certain genetic treatments that are in trial phases for cancer right
Yeah.
now.
And you're referencing like CRISPR technology?
Right.
Yeah.
So if we can, leveraging this, we can identify these single nucleotide polymorphisms, or these genes that are very bad. And potentially in the future, we can use some of the existing technology or some of the emerging technology, like nanoparticles, for instance, that we can pack signaling factors into, like microRNA. And we can turn off cancer genes, or we can modify sickle cell and actually have a very viable treatment to improve the life and the quality of life, not just the life alone of people that are affected by sickle cell or who get cancer. Leveraging this data, I think we're closer now to a cancer cure than we ever have been. And one of the questions of interest for us is, using this data, can we find genetic signatures to where if people cannot exercise, can we have a drug that can be repurposed that will mimic the effects of exercise so we can get at least similar benefits? And that's something that would be very powerful in a clinical setting. If you think about people who are undergoing amputations or major surgeries, or even astronauts coming back from space after an extended stay where they don't functionally have the ability to move around yet because you're at zero gravity for so long, and then you come back to negative 9.81 Newton meters per second of gravity on Earth, and you don't know, you're bedridden for a while until that adjustment happens, until you can gain strength to get back into physical exercise. We could have an exercise mimetic in a pill form or in a lipid nanoparticle injection, similar to how the mRNA vaccines were developed, that could give us some of those benefits so we could improve performance or get back to a healthy state quicker.
Fascinating stuff, man. Well, would you agree right now, as best we know, that exercise is the most potent medicine, multi-system, we would say. Certainly not to discount the appropriate application of pharmaceutical technology, um, but man, if you could encapsulate uh, or recombinate the effect of exercise, on health and our physiology, that would be the absolute most powerful, helpful medicine available to us. Would you agree with that?
I do. I mean, when you really dig into the disease literature in particular that have participants with a disease undergo exercise, it is universally helpful.
Hmm.
If you have diabetes, it's helpful. If you have cardiovascular disease, it's helpful. If you have COPD or any other pulmonary type disease, it is helpful. If you pre-hab before you undergo surgery, your outcomes are better.
Right.
If you are doing any type of exercise, it's better than none, even if that's just tracking step counts. And all of those seem to go a very long way as far as longevity and resiliency as well. And we're seeing that over and over and over again in our data. Um, my main mentor, Dr. Virginia Krause, published a paper 2 years ago, 2022, on causal determinants of longevity using a 1,000+ participant cohort and the ability to do individual activities of daily living. So if you think about that, we can classify that as physical activity. Those are things like cleaning your house, taking the garbage out. That is highly predictive of 2-year longevity, 5-year, and 10-year longevity after a blood draw for those with that are older in age.
Hmm. Wow. Well, in my read of the literature is just one metric that's been popularized in recent history, that like when you really look at the associations, it's quite striking, right? When you see the relationship between VO2 max across, that's men and women 18 and up. If you look all the way up into the 80s, the higher that VO2 max is, the more likely someone is to be alive in some subsequent year later. Um, in one study in JAMA, I was in 2018, that comes to mind as, I believe it was like 120,000 participants, and the difference between the bottom 20th percentile and the top like 2%, so 98th percentile and up, when you take VO2 max and you look at the hazard ratio, the likelihood of survival across a decade or more, the difference between like the lowest quintile and the upper one, but particularly when you get up into like the top 2%, I mean, dude, it was like 400% difference. I mean, 400, 4x, like that's wild. And I mean, just to kind of put that into perspective, the difference between an individual, let's say, with like a 45 or a 50 BMI as compared to someone with 25 BMI, let's say, that class 3 obesity versus in that sort of normal range, 23 to 27 BMI. I think that the hazard ratio is close to like 2. So maybe 100%, 150% difference. So certainly still meaningful, but like that's interesting because it's like, man, cardiorespiratory fitness as measured by VO2 max, which is one of the key assessments that we do in the Phytomics Fitness Lab. While we're so adamant about that biometric is, its importance for health span.
Right. I think it's also relatively important, even if your outcome goal is not to have a great VO2.
Right.
Your outcome goal is to be the size of Ronnie Coleman when he's deadlifting 800 pounds for a two-piece.
2 weeks out from the Mr. Olympia, I might add.
Yeah.
Legend. Legend.
But cardiorespiratory fitness has recently been identified as part of the process that also improves hypertrophy outcomes. And if I'm not mistaken, did you review that article for
That,
reps?
yeah, well, so originally the Thomas paper, yeah, I just shared a research insight on that and revisited it, and what a striking study. And you mentioned Stu Phillips, like he was on there, um, and that was for some reason I don't think that's gotten as much attention. What they actually found, I found that to be really striking. How the 6 weeks of so just real quick, um, the within subject design, right? If we were all in this study, we would have all done, you know, well, back up. So two legs, one of our legs, we would have done 6 weeks of aerobic conditioning, while the other leg wouldn't have done anything, at like 50% of the peak work rate measured during a VO2 peak test. So probably right around like zone 2, if I were guessing, heart rate. So nice moderate pace. Not you could probably talk during it. Pretty easy bout, but noticeably exercising, right? And 3 times a week for 45 minutes a week, the leg that was exposed to the aerobic conditioning would have done that for 6 weeks. The other leg wouldn't have done anything. 2 weeks of rest after that, followed by 10 weeks of resistance training for both legs. So both legs did the exact same 10-week resistance training protocol. The only difference was one of our legs, we would have done the 6 weeks of cycling, um, 3 times a week for 45 minutes. And they collected biopsies before the aerobic conditioning portion, after, and then after the resistance training portion. And was really surprising, that they found, I think at least, the magnitude of difference. Just looking at percent changes, it was 14 subjects, I believe, in the study, but it's within subjects. So like controlling for genetics, diet, like it's relatively strong study design. And so, yeah, the leg that did the aerobic conditioning prior to the resistance training, we saw about 40% increase in the type 2 fiber uh, cross-sectional area, as compared to, I believe it was about 15% in the leg that did not perform the aerobic conditioning. And I believe it was like 15% in the type 1 fibers versus like a few percent change in the non-aerobically conditioned leg. And that's interesting. And they also found, right, that the capillary to fiber perimeter exchange ratio and capillaries per fiber tended to be a little higher in the aerobically conditioned leg, so better blood flow delivery, perhaps even better activation of satellite cells, which you touched on before, because that did seem to be based on correlation. The more the larger the increase in satellite cell content seemed to correlate with the change in fiber cross-sectional area of the type 2 fibers. But there was also a relationship between the capillary to fiber perimeter exchange ratio and change in type 2 cross-sectional area. So that's interesting, right? That there's probably a symbiosis, you could say, between conditioning at the appropriate dose intensity, and resistance training that are conducive to promote muscle growth. It doesn't have to be just either one. But without going too far, down that rabbit hole, I think that this was fascinating. We are 2.5 hours in. Uh, we could definitely look at if you would be so gracious to honor us with follow-up um, conversation, because my anticipation was we would have made it through a couple other of the topics that we had briefly talked about there before we got to connect. I think there's much more in store. I think what I would like to close with is to give you the opportunity, Dr. Vann, to tell us like what you're most excited about as far as research and what you're working on now, and like what you're going to be focusing on. You know, with all the you touched on one study today. You've done several now that are really interesting. What are you most excited about? Like, what are you wanting to focus on here coming up in the next, let's say, 5 to 10 years of your research career?
So my larger interest, we know with exercise that the bulk majority of the work is done through striated muscle, both skeletal muscle, cardiac tissue. But what we universally see is every other tissue in the body benefits. And that's fascinating to me. So one of the central questions that I have is, what's causing that? So I've gotten into the realm of extracellular vesicles and signaling from extracellular vesicles to other tissue types. So that's really going to be the focus of my work for the next 5, 10, 15, 20 years. Using various models, cell culture, cell co-culture, creating tissue engineered systems where we'll be able to share circulation but have directional flow from skeletal muscle with labeled extracellular vesicles with what's called a green fluorescent promoter or GFP. So we'll be able to see the localization of extracellular vesicles to different tissue types. So the goal is to, one, identify a better surface marker for muscle-based extracellular vesicles. I don't believe that the current most widely used surface marker is the A+ job. I think it's good, but it's not ideal. So currently that marker is alpha-sarcoglycan. And if we think of the muscle cell structure, it's part of the dystrophin sarcolemmal complex, but alpha-sarcoglycan sits outside of dystrophin, more towards the basal lamina. So there's a little bit of debate on, is that really muscle specific? Could it be fibroblast related? Could it be one of the any other 8 to 11 different cell types in the muscle milieu that are pushing those extracellular vesicles out? And extracellular vesicles by themselves are very interesting because every cell pushes them out. It's part of the general cell cycle. So they pull things in through the membrane. If they use it, great. If they don't, they package that up through the lysosomal complex and they push that out. But cells also undergo cell death or apoptosis or apoptosis. And with that, you have apoptotic bodies that are released or large extracellular vesicles, and generally the field focuses on small EVs. I think there's utility in really focusing on the whole gamut. There's small, medium, and large. And the large EVs carry small EVs within inside them. So it's quite a fascinating area. It's relatively new. And with that, we also know that they carry a slew of different cargo, proteins, DNA fragments, histones, RNA, microRNA, small RNA in general. So they could be having very dramatic signaling effects throughout the body. So I haven't followed up on this experiment yet, but I did a quick pilot just to see, if extracellular vesicles had an effect on skeletal muscle cells. So I took C2, C12s, I grew them up in standard conditions, and then I starved them for 3 hours as an acute starvation. I had isolated muscle-specific extracellular vesicles and treated the starved cells and saw a very nice, robust increase in PI3K AKT expression at the protein level. And then I did a chronic starvation where I starved them for a week and did the same thing and also saw a very nice upregulation of PI3 AKT signaling. And if we think about PI3 AKT in general, that signaling cascade is involved in so much. It's involved in cell growth. It's involved in cellular senescence. It's involved in a lot. So it was a nice target just to get a confirmation that, hey, these are actually having beneficial effects. So part of the question is, well, if we take these muscle-specific extracellular vesicles after an exercise intervention and we treat other cells, say we treat cardiomyocytes, where we can program in treating them by themselves to give them a phenotype, we can program in with electrical stimulation an infarction. So now we have damaged cardiomyocytes. If we treat with extracellular vesicles, can we repair those? Can we repair the endothelium in the blood vessel linings? Can we repair the fibroblasts that make up a good portion of the extracellular matrix? So those are part of the initial questions before, and getting that all situated and doing extensive research in cells before taking that up to rodent models. And then expanding past extracellular vesicles using lipid nanoparticles as a means of potentially developing targeted therapies. So it kind of all plays together. And then, of course, I am highly involved in a lot of other studies where we're using exercise interventions to really identify genetic determinants of not only phenotypes, but we're also interested in identifying genetic determinants of whether or not someone's going to adhere to an intervention. And that's not just exercise, that's also dietary related. And it would also have crossover to, if you're prescribed a drug, what's the likelihood that you're actually going to take that drug? So leveraging all of this data to get at those type of answers, that's the trajectory really over the next, I guess, until I die.
Okay.
Because I'm also the type that will probably die in the office.
Well, hey man, we're gonna believe that you're gonna create this extracellular vesicle cocktail that if you start feeling weird, you just take a shot in the arm, buddy, and you'll be regenerating all kinds of things. Grow a tail, maybe breathe fire. Those are the sorts of things that I think we should really focus on. Well, dude, hey man. So extracellular vesicles, honestly, that would be like a really cool follow-up conversation, teaching us about your work in that area. Well, okay. Tell me, Dr. Christopher Vann, where can people find you, your work, like keep up with you?
That's a great question because I am a rarity these days. I do not do social media at all.
Okay.
I have an Instagram. I think it's VANNCG1, but I haven't posted on there since 2017.
Okay. Got it.
I also have a Twitter or an X account under the same, but I've never posted on there.
Okay.
I guess I just troll people, but
Fair enough.
yeah. So I guess the best ways to keep up with what I'm doing, I have a Google Scholars profile where all of my work is available. You can also search me up on PubMed, or if you just type in Christopher G. Vann into Google, you'll get my Scholars profile, where a lot of my work is also kept up with. So those are probably the most accessible ways.
Fantastic, man. Well, and in the coming future here, we definitely want to bend your ear and listen to your soothing tones. Teach us about science and all of the research And with, our work with Phytomics, aiming to help bridge the gap between this sort of research and actual implementation from lab to life, if you will. And so kind of translating that. And so, hey, man, this was a blast. Uh, Excited about more of this in the future. And I think, yeah, next time we can dig into extracellular vesicles. And then there's like a virtual lifetime of further conversation as we continue, doing more and more research and learn more. Dr. Daniel Lawson, do you have any
What's up, brother?
concluding remarks, concluding comments, questions?
Yeah, I mean, I am burning with so many questions in my brain right now. We could do this all day, particularly with some of the omics and tying that into like lactate. I think there's a very interesting deep dive with that, and would definitely like to get more into the ripping heaters and muscle growth.
Yeah.
But yeah, I think we can say that this was great. I guess one question, if it's possible, if we were just to zoom out for some people that make it to the end of this this podcast, right? For gym bros like myself, right? Like, what's something from this talk and from omics that somebody could take away and utilize that in their own training? What's maybe like a couple of one or two like golden nuggets that you would give to that individual?
So I think really there, it's be willing to try things, because we talked a lot about heterogeneity of response and how some people don't respond to a given intervention. But we have to keep in mind, contextually, interventions in the lab are created to elicit a very specific effect. And if we don't see fiber cross-sectional area changes, maybe it just was that intervention that didn't work for you. Maybe you try something else. Like, there are some people, and we've seen it with Cody's study in particular. This comes to mind because we did a questionnaire with those participants often. One of the questions was, do you feel like you could have done more? And, full disclosure, Cody and I ran his study on ourselves. We did 3 different iterations of it. And during week 6 of the 32 by 10, I very specifically remember a point in time where I passed out in the gym, laying on a bag in my 2-minute rest period. I had a 1-pound bag of Sour Patch Kids next to me, a bottle of Mio Energy, and then my water bottle. And I woke up to Cody standing over me, just going, "Yeah, are you done?" And I reached over and just grabbed a handful of the Sour Patch Kids, downed them, rinsed it down with some water, and then took the Mio Energy straight to the face without diluting it or anything. And I did my last sets. And we were just angry. And we did DEXA scans on ourselves. And interestingly, from start to finish, we both grew a lot of muscle. What would probably end up being statistically significant. But with the participants that we had, some of them grew a lot more, some of them grew a lot less, and some of them participated in my high load studies that were at lower volumes. And with that high load intervention, they grew significantly more than with high volume. So different things are going to work. So you have to be willing to try. And if you're working with a professional, which in a lot of cases I think is a really good idea, there's a trial and error that does go into that while we're trying to figure what's working out best for you. The clients that I've worked with where I have the best results, those results generally start trickling in after about 6 months of working with them, because we're trying things and we're getting things dialed in. And on the dietary side, as a generality, a lot of people use like the Harris-Benedict formula or the Mifflin formula to generate a base number of calories that you need to sustain life. But the precision with that comes in the longer that you work with someone, because you start to learn and identify especially with how, at least how I track, I think Cody and Dan still track in similar ways. You identify certain energetic responses to loads, to interventions, to aerobic exercise. And then I also collect data on what they do throughout the day, because in today's society, most people aren't going out and laying asphalt for 12 hours or roofing for 15, where you're burning a ton of calories. Most people exercise for an hour a day and then they sit behind a desk for 9 hours. So those formulas are great, and we have standardized values for activity expenditures, but those are really geared towards modifying the total daily expenditures. And those might not apply to today's society where, you know, you're sitting there for 8 hours. Maybe it's not good to multiply your total calories by 1.3 for light activity.
Mm-hmm.
Maybe you need to dial that actually back to maybe 1.15 to see progress. And in some cases, maybe you actually need to dial that up to 1.6 and you're still losing weight or gaining weight. So part of it is getting all of that dialed in. And that happens a lot through trial and error. I think that that's probably the main thing that I think is the best takeaway, because the genetic determinants and everything, that yes, they certainly play a role, but a lot of the minutia that goes into developing what you're going to do for the day also plays a huge role.
Yeah, there's a lot to unpack with that, but one of the things that my mind goes to and has been a thought that I've had as a practitioner and as a scientist in this area, is I wonder, right? You mentioned that like a high volume resistance training program worked for some and not others, but some people responded better to the high load, lower volume, within a certain time frame based on these measurements, right? That actually elicited a greater response in muscle hypertrophy. One of the things that I keep wondering about is like, does that change though? Just because the one time you did a high volume training intervention and it didn't work very well, well, maybe 6 months to a year later, you run that program again and all of a sudden it works, right? You talk about like high volume. I remember my first, I think it was first year of my PhD coming out of the master's. And a lot of this was based on Dr. Cody T. Hahn's dissertation was high volume. What's the limit? So I was blasting volume. I remember when I was using a training app to log these metrics. At one point, it was up to, I think, somewhere between 50 and 60 sets per muscle group. And we had to split that up. So my training partner and I, shout out to Sergio for going through that with me. We had to do 2 sessions a day, and it was COVID. So we had, the campus was shut down. It was kind of the ideal conditions where I could just get up, eat, sleep, do what I want, focus on training. And I could handle those high volumes of training. And, we would split that up into 2 sessions at one point because it was like 45 minutes to an hour each. And just anecdotally, I didn't do DEXA. I didn't do anything like that aside from, I think an in-body home scale. But the pictures, and when you have a decade of experience, I've been doing this since I was like 13. 8th grade is when I got under a barbell and have consistently done that. That's, that's almost 20 years, of in the trenches training. Right. And then working with hundreds of people, athletes to Gen Pop, right? Like, when something changes and when it works, right? That's just the coach's eye. And I remember getting up to those high volumes and some of those pictures that Cody has taken of me or that I've posted on social media from a couple of years ago are disgusting. But I remember, getting up to that extremely high volume and working up to that, I noticed it was like dramatic changes. And I wish I had some sort of quantitative measure of that. But if I were, and the point with that is at one point, Sergio and I were like sitting at the gym and we were about to do we were doing our PM training session for one of our lower body days. And we were about to do set 8 of our rear foot elevated Smith machine split squat. for a set of like 8 to 10. And I just looked at Sergio and I was like, I don't want to do this. He's like, me neither. And that's where we tapped out. So we're like, we're done, right? Like we finally hit a ceiling when every muscle group was being blasted with, again, 50 to 60 sets. I made a post on it so I could find that number somewhere on my Instagram. You know, that was kind of the conditions were set for that, right? One is we had time to focus on training, nutrition, sleep. Stress was relatively low. But if I were to do that now, you know, I don't know that I would respond as well, maybe, right? But I wouldn't be able to do it. I think I can't commit that much time, one, but even if I did, I don't know that the results would be as good, right? I don't know. And I've been doing a lot lower to moderate volume right now. I haven't really been increasing set volume within the tactically thick operation phase 1, which you can find on the Fidomics training app. Shout out, plug in that. You know, what I've been finding right now that has worked very well for me personally is I'm doing like 3 working sets for my, you know, my, I guess, quote unquote, hypertrophy exercises within that current plan. And I'm just focusing on, you know, really good technique and trying to increase the load on the barbell or the dumbbell or on the weight stack machine, right? Plate loaded machine, whatever it is, just when I can try to increase the weight or, you know, again, this is why tracking things is so important. You touched on that, Chris. Why, yeah, it's so important to track things because I can look back at historical data within an app and try to do a little bit more, right? It's like, okay, last time I did this weight on this machine for this many reps, let me try to get 6, right? And might be getting a little bit off topic here, but to kind of circle it back, because I found that has been working anecdotally, right, very well for me right now, is just kind of keeping this, you know, and volume can be defined operationally different ways, right? So set volume is not increasing, but I am trying to, I guess, quote unquote, increase volume, right, by doing more weight or more repetitions or both. But I guess the kind of like circle back to that is I don't, you know, I think it changes, right? Like what program works for you now might not work for you later, right? And that's part of that trial and error, you know? And so I think there's a lot of factors that play into that, but I think it changes.
Yeah, no, I agree. And for those that do work with coaches, I also think it's really, really important to have consistent communication with your coach. If you're noticing a problem, if you happen to have a tight hamstring out of nowhere, even it might sound minor, it's worthwhile to let your coach know because modifications can be made. I also think it's really important, and you touched on this a bit, training is only a small part of the equation. The nutrition side is a big portion. Your lifestyle factors are also a huge portion. We know that little things like how things are going, if you're in a relationship, those affect your ability to train. If you've had a death in the family, if you've had a birth in the family, all of that factors in. If you live in a dangerous neighborhood and you're just sitting there stressed because you want to move, but you can't afford to move, that's going to affect that. Your stress loads, your extracurricular activities, how much ethanol you consume, if you partake in legal cannabis or maybe even illegal cannabis, that can all affect things. There's a whole lot. And one thing that I really truly advocate for, particularly athletes that I work with, and even Gen Pop that are on dedicated programs for long periods of time, I always advocate the 1 month a year taking time off and doing what you feel like doing. Because usually, particularly when we get to a high level, our fitness goals aren't necessarily tied to I want to be bigger or I want to be leaner. They're often tied to a certain outcome. And for a long time, you know, mine were all growth related for a long time, but there was a portion of time where I was working towards a 600-pound squat and I never quite made it there. I made it to 550. So, and for the record, genetic wise, I am a heterozygous positive for ACTN3, what's regarded as the muscle strength power gene. And that doesn't, I'm relatively strong. I mean, realistically, when you look at it, if you can squat over 315, deadlift over 315, and bench over 225, you're roughly in the top 5% of humans, strength wise, for men. And the other thing that's interesting is I don't look at myself as being strong. So, all time numbers, as far as raw powerlifting, my best in competition was a 500 squat, 500 deadlift, and a 365 bench. And at the time, that was really good. But comparing yourself to the world's elite now, particularly in the weight class that I was in, which was 83 kilos, you're looking at Russell Orhe and now Taylor Atwood, who are taking those numbers for like sets of 12, 15. And it's like, okay, I'm weak. And a lot of times what we see is we see everyone, the influencers on social media, and there's nothing wrong with that. I love watching those guys and gals. But it's important to realize that what we see is a snapshot. And those are the elite of the elite. They're the top 0.01%.
Mm-hmm.
And it's not necessarily a fair comparison. The comparison, I believe, and this gets into the psychology of things. I think the comparison, whether it's bodybuilding, whether it's powerlifting, yes, if you're competitively powerlifting or bodybuilding, you want to win. Or at least that's the way I am. I want to win. And that's in everything that I do, not just athletics related. But the real comparison is, did I get better today than I was yesterday?
Mm-hmm.
Am I doing better in every aspect of life today than I did the day before? I think that's the more appropriate comparison, and that's harder to get to.
Mm-hmm.
Mm-hmm.
Yeah. Well, hey, that's a good one-liner to wrap us up, man. I think that this was really good, and Dan, it was good to conclude with some practical applications. And the theme of the response there was it's important to be open to try new things, and if you didn't respond really well to a given training style, dose, program, it doesn't mean that you won't respond well to another, framework, another iteration of that. And Dan, to your point, and tracking that, like having, and that's, with Fitomics, like that's part of our mission is testing. Like we want to be able to quantify both physiology and then just practical outcome variables like body comp and VO2 max and strength improvement over time in order to be able to determine what will work better or worse, right, for someone, and predict how someone will respond with better probabilities. But tracking and being willing to try different things, and in one season of life, what worked, given the constellation of factors that existed then, may not work again in the future, and so having that sort of data and that monitoring process in place and working with a coach, which is what we really aim to do through Fitomics, coaching is
Mm-hmm.
help individuals put these pieces together and have a logical plan to follow, and someone that can be there to modify, be supportive, share resources, those sorts of things. I think, in the future, man, it would be really cool, right, if we were able to, in the comfort of one's home, collect some saliva, a little blood, maybe a little urine, stool. I know, Dan, you're big on collecting people's stool and storing that in your freezer. But stool,
That's right.
so we get these biological samples, and there's a way to get information from that day, like, and make some useful decisions, based on that, as far as nutrition, supplementation, and exercise, right? Like that'd be super cool. And omics, I think, is best we know now, like a way that we could actually move in that direction to get that sort of detailed information that's actionable, that can help people to right, train and eat smarter and get better results. And so a lot of the things that we're doing now with like the coaching and like Research Labs Insider, opening access for research and informational purposes for folks to be able to test various biomarkers that could be influenced by their nutrition and training strategies, other things that they could use to make better decisions, right? So, hey, man, this was fun. Thank you so much for your time. I'm confident, God willing, we will be able to host you again and connect with you, at least from this remote. You're in Durham, right?
Yes,
Yeah.
roughly.
So perhaps in the future we can actually snuggle up next to each other with some essential amino acids, maybe a little creatine monohydrate, and we can all, hang out in person and do a recording there. But I think that there are a lot of fun and exciting conversations ahead on the science of fitness. So signing off, we have Dr. Chris Vann, Dr. Daniel Lawson, and Dr. Cody Hahn with Fitomics. God bless you all. Here's to your fitness. We'll see you next time.